to do is to go full screen. Looks like all my lights are on. And here we go. The outcome of fertilization, of reproduction. The terms development and inheritance are kind of familiar words to us, but what do we really mean in biology in A&P when we talk about development and inheritance? Development here, the gradual modification of anatomical structures and physiological characteristics from fertilization to maturity. Now, that means, to me, we start out as a single squishy cell, the fertilized egg. Now, I want to emphasize, a single cell is still the beginning of each of our lives. Although we're only really alive and individuals as a multicellular organism with trillions of cells, we still go back to that single cell stage to begin our life. From that point, if you want to select a certain point in development, like, for example, live birth, then you're talking about how that single cell makes the multitude of cells and begins work on all of the tissues, organs, and organ systems to produce an eventual mature adult human. That's development. Development actually will extend beyond live birth through stages of development, so we might name infant, toddler, adolescent, and then once we achieve sexual maturity, adult development, maturation, and eventually aging and death. Inheritance is the idea of what are those principles that allow us to understand the transfer of traits from parents to offspring. Now, in human beings, our amplitude for reproduction is very limited. Human beings only reproduce one way, sexually, with two individuals of different genders. Turns out this is a very, very limited scope for reproduction. My initial work was with plants, and I know there's a plant that I studied which grows basically as a weed. It's truly a wild plant in this area of deciduous forest that's changing gradually to savanna and prairie as you go west through Missouri. That plant could reproduce by fragmentation. So if you cut off a branch, it would root in water. If you basically bent this branch down and just put a brick on it, it would grow roots into the ground. That's generating a new individual by clonal reproduction, by vegetative reproduction. It's an exact genetic copy of the, quote, parent plant. That same plant would produce flowers seasonally, and because it was a perfect flower, meaning it had male and female parts, it produced both megaspores, which is the plant name for egg, and microspores, which is the plant name for pollen. That plant could be a sexual reproducer by fertilizing itself. So its own pollen on its own stigma would produce a viable seed that would germinate. It would be genetically different from the parent, but it would have only one parent, even though it's a sexual product. Finally, that plant, in opening its flowers, it matured the female surface before the pollen opened. So in its population, if there were pollinators like bees flying around, they could pick up pollen from another plant and bring it to the stigma of the parent plant. And now you have the typical kind of sexual reproduction that humans are familiar with, two-parent reproduction. So here's one species of plant that has basically three different ways to reproduce. So if you have a great genotype and the emphasis is on rapid reproduction of your single genotype, vegetative reproduction is the way to go. It's quick, it's convenient, it's great for colonization. If your seed has a pappus that floats on the wind, or a wing like a maple leaf that gets blown, a maple seed that gets blown on the wind, and it lands by itself in a new place, it can basically germinate, grow up by fragmentation, can produce multiple individuals, and by self-fertilization can produce sexual offspring. Humans can't do that. We reproduce only in this one way. Once the fertilized egg is achieved, cell division occurs with the first few divisions of life. We have a term cleavage that refers to the mitotic divisions in a fertilized egg. So the first cleavage would produce a 2-cell stage, the second cleavage a 4-cell stage, and so on. Now, at first, those cells all look the same. It's occurring so fast, and you're building up to that 32, 64, 128 cell stage, and it's just a ball of cells. Rapid cell division. But before long, those clumps of cells begin to fold and bulge and extend. Cavities develop inside them. You begin to see differences, so that it's no longer just a ball of cells. It definitely has an end with a lump and an end with a furrow. This is the process of differentiation. Those cells are specializing. They quickly become a cell in the head end or the tail end of the developing embryo. And that's a result of differential gene expression. Genes are being turned on to produce that characteristic embryology that's going to lead to a human embryo. Fertilization differentiation is actually the differential gene expression that produces new cell types. By that I mean tissues that will combine into organs as development runs its course. Fertilization or conception is when development actually begins. That fertilized egg is the first cell of life. And finally, we get a good picture. I've made the point before that compared to the egg, the sperm is a midget, although a very fast midget. So this sphere right here is an egg. Now this is probably from an in vitro fertilization, where eggs that had been extracted are being fertilized in a petri dish by introducing sperm, but you can see they're absolutely covered with the sperm, this huge egg. And here is an individual head of a sperm and its long tail. So you can see what I mean about this cell being gigantic and loaded with resources, all the organelles, all the energy, all the chemical additions necessary to maintain that intracellular life are here in the egg. And this sperm is basically a half nucleus. It's a precise half nucleus. One number 1, one number 2, ... one number 21, one number 22. And either an X or Y. But that's the main thing that the sperm is delivering in its contribution to the fertilized egg. So from this stage of fertilization, and showing down here below, copulation has occurred ideally because the sperm had the ability to swing up through the uterus, find the uterine tube and swim all the way down here, so that when ovulation occurred and that positive current sucks it up, it begins the egg's journey. The sperm are right there to fertilize it immediately. I'm hearing more and more, shorter and shorter spans for the average survival of an egg. Most people are saying 24 hours from ovulation. So early fertilization produces that fertilized egg, and immediately you begin taking the size of the egg and dividing it up based on rapid mitosis. Now, normally in my body, if a mature cell dies, then a cell near that space will activate and divide into two daughter cells that are about half the size, and then they will go through a growth stage and reach that full mature size before they divide again. The fertilized egg is not that way. So this first division, you can see, basically divides the two cells. Here are the two half-sized daughter cells. But instead of waiting for growth, they divide again. Here's the 4-cell stage, and this is occurring within a couple of days. We're already at the 4-cell stage. That means a couple of things. The presence of a fertilized egg and chemical signals are coming off of this egg that is signaling the whole female reproductive tract that a pregnancy or a potential pregnancy has begun. As you go 4, 8, 16, 32, 64, the early embryo is just a ball of cells. When we get to the point, we can no longer conveniently count them. We call that the morula stage. It is a solid ball, and the breakdown between the morula and the blastula is when it reaches a size where diffusion can no longer get sufficient oxygen into the center, cannot transport sufficient nutrients into the center, or transport wastes out. So what this particular picture, morula to blastula, shows is that at some point these cells begin to migrate and differentiate into a cavity. You can see this single layer of cells on the outside called the trophoblast, surrounding a cavity, the blastocele. The term coelom, C-O-E-L-E-M, refers to an internal cavity surrounded by cells. But also, in addition to forming this different kind of sphere, this hollow sphere, the blastula, you start to see differences between the surrounding cell mass and the intercell mass. Differentiation is basically producing a protective layer and an embryonic layer, meaning the cells that will become the actual offspring. So by the time you get to day 6, day 7, the trip down the uterine tube and into the top of the uterus is made. And the chemical signals coming from this embryo will cause it to attach to the endometrium and activate a pregnancy stage. This process of attaching and burrowing in, part of it is burrowing in, part of it is the activation of the cells in the uterus that actually grow around and enclose a portion of the embryo. So this is shown here, the blastocyst from the outside, and the trophoblast kind of in profile, showing its contact at about a week with this renewed endometrial lining, basically the effect of menses and the regeneration, the luteal phase of renewal of the uterine lining, has led to a condition where these two cell masses react to one another. You can see this growth. It's kind of like a root-like extension into, and then the growth of the endometrium around. You can see developing villi or tube-like projections that are moving into the uterus and securing attachment, also initiating the development of what will be needed. Now, what do I mean, what will be needed? Well, we're constantly continuing to divide these cells. So as we divide them and we get a bigger and bigger cell mass, we have that same problem. A mass of cells, diffusion can only get oxygen and nutrients so far, and inner cells will die. And so as this increases in size, the elaboration of the embryo in this portion and the elaboration of secondary tissues in the uterus, notably the cavity that will enclose it, the fluid that will fill that cavity to keep the embryo moistened and keep it protected, and the connecting blood vessels of the placenta will all begin to develop. So this is an ongoing process. You can see it begins as an embedded group of tissues. The uterus has grown out completely enclosing it, but it does have the characteristics that we see in sperm egg reproduction. This is the blastocoel cavity, and on this stalk we see the development of a special membrane called the amnion. This is why it's called the amniotic egg. And this particular stage of development, we have very, very few cells that are contributing to the embryo itself, but a scanning electron microscope micrograph of a embryo. Now in this case, this is a monkey embryo. You start to notice how this basically still spherical or slightly oblong embryo has an uncountable number of cells, but definitely is developing a shape of its own. This is the tail end. This is the amniotic cavity that enclosed it with fluid. These somites, and you can barely see them here, 1, 2, 3, are segments that will eventually become the vertebral divisions of the spine. This is the axis of the spinal cord and a so-called neural plate. This is the future head end. Now, although this is a monkey embryo, one thing that struck people who studied embryos and their development was how similar the development in mammal embryos are in the beginning stages. So the development of this axis, a kind of double flap here at the head end, this is the end that will become the head, and a sort of a single tail-like protuberance around a rift. The tail end is very, very similar. It kind of doesn't matter whether you're talking about a human or a monkey. It doesn't matter. In fact, if you look at a lion or a sea lion, at a bird or a snake, at the initial, at a bat, the initial development of the embryo is very, very similar. So back about a month, what do we have? Here we have this embedded amniotic sac filled with fluid. But we see a strand beginning here, and it basically is sort of like a fish hook shape. There's an enlargement on one end, which is the head end, and a long extended tail and division into definite segments. That is accompanied by this stalk. You can see the connections to the uterus and the ability to transport material in and out of this embryo. You see a yolk sac, which is initially in development larger, and as that yolk is utilized for its stored energy, it becomes smaller and smaller and eventually disappears. But however, that yolk does serve as nourishment for this small group of cells. As the placenta is developing its real exchange area, by that I mean the placenta capillary plexus will be next to a capillary plexus from the mother side and create this exchange zone. The blood flowing through the mother side of the uterus is going to be delivering oxygen and nutrients and picking up waste in this zone. And then the excess oxygen and nutrients are going to be absorbed when the placenta is developed and transported around the embryo to support its development. The early beating of the embryo heart, the contraction of that muscle, is what's going to propel the baby blood around that side. But it's important to realize in this development there is no direct attachment, no flow of mother's blood into the embryos or baby's infant's body. As we look at a 4-week fiber optic view, definitely see kind of an odd but distinguishable head area with an eye spot clearly developed. These are called pharyngeal arches, and those arches actually represent the openings that form gills in fish. in amphibians. Amphibians are born in a swimming form with gills and later transition to air-breathing lungs. And those develop in human embryos. Now, because we aren't born with gills, they are grown over and replaced by genes and gene expression that came later in evolutionary history. Here is the ear region where hearing will develop. And along here, see this segmentation, or what are called somites. Those are the individual segments that will become the vertebra. We do see an arm bud, and we do see the beginnings at a month of a leg bud. But one of the things that's really dramatic at a month is this long, luxurious tail. So here's where the leg attaches. And behind the leg attachment, there is this long, beautiful tail on the human embryo. Now, whether it's a bat or a snake or a chicken, at a month, it all looks about like this. At 5 weeks, we're going to be looking now at the entire uterus. Now, as this develops, we are expanding the uterus. It is growing with the development of the embryo itself. The embryo is this crosshatch piece right here. All of this is accessory tissue. The accessory tissue and the fluid of the sac that cushions it. But as weight begins to develop and more accessory tissues begin to develop, this uterus begins to expand. Now, here is the development of that exchange zone that I called the placenta. Blood vessels in the wall of the mother's uterus are bringing in resources in the blood, and basically oxygen and nutrients are enriched in this region. They are picked up by the capillaries on the baby side, and as the heart begins to beat, circulated through the baby's body and returned to this position. This is where wastes would be exchanged and more oxygen and nutrients picked up. So over here on the mother side, except for circulation of the blood, which is supporting the development of the embryo, not the operation of it, but just the development of those new cells and their support, all of the mother's organs are doing double duty. Oxygenation and the exchange, the removal of carbon dioxide, nutrient delivery, and waste extraction. So the mother's kidneys, the mother's digestive tract are providing all of those services that will be independent once the baby is born. Here we have a 8-week view at 2 months, and we definitely start to see that that generalized form that was present at 4 weeks has changed into something that actually does resemble a human. So here we have the placenta and the umbilical connection through the navel of this developing human. This amnion continues to surround it, the amniotic sac, and the fluid which hydrates but also protects the embryo within its development. It clearly can see an eye, an ear, bud, the forelimbs with fingers, the hind limbs with toes. And you can see the remainder at this point of that tail. What the embryo is doing is it's basically growing to match it in size and surround it. And so basically the tail is just incorporated into the spinal column. And the coccyx, which used to be the extended tail, is the development of that is turned off in humans. We are members of a group called primates, and you know that many primates have wonderful long tails. Those that are still brachiators and spend much of their time in the trees sometimes have what's called a prehensile tail, a tail that can wrap around a branch and kind of act as a fifth hand when you're up in the trees. In one group of primates that evolved fairly recently compared to the origins of the group, primates began to appear without any tails. Tailless primates are called apes. And the genes for tail production were not removed, they simply were turned off. And so as a representative of that group, the tailless primates, humans have no tail normally. So proceeding on, here we are a couple of weeks later. The baby is shown turned at this point, but you can definitely see the expansion of the placenta and attendant tissues that are attached to the now thickened endometrium, the so-called decidua parietalis, as in parietal layer. Now, decidua is an interesting word. It is the root of what we say is deciduous, which means that there is a point in development where the connection between two things dissolves and the part that was connected falls off. We refer to our broadleaf trees in this region as deciduous trees because in the fall, those firmly attached leaves, you know, you go up in the summer, grab a leaf and pull it, it's firmly attached. It's actually solid attachment. But in the fall, a layer called the abscission layer dissolves so that eventually those leaves, literally from the weight of their own leaf body, fall off. The same thing is going to happen here. This is a firm attachment during development, which it becomes more and more elaborate and extensive as the number of cells that are being supported increase. But as we approach the end of gestation and we approach the time of delivery, which is called parturition, this tissue begins to decay. So when the baby comes out and the umbilical cord is cut, typically there's a cessation of labor contractions and a moment of rest, but then a resumption, and this entire tissue that was elaborated for the support of this fetus becomes deciduous and separates from the wall of the uterus. Contractions force it out of the uterus, and it follows the same delivery path until it is expelled from the body. And it is for part of the kind of cleansing and healing processes that will allow this uterus to contract and in time resume its smaller form and resume its cyclical, basically resume its role to support the cyclical maturation of eggs, which will resume after delivery. So here's 3 months. Definitely see human appearance with fingers and eyes. You can see the brain in profile through this largely translucent covering of the skin and the skull. Those bone plates have not really begun to develop to any extreme extent. You can see the so-called central sulcus and the longitudinal fissure dividing those up into lobes, and definitely a human appearance. At this stage, you would begin to get movement as muscles are developing. The second trimester, or months 4, 5, and 6, the fetus grows very much faster, and it outpaces the growth of the placenta. The third trimester is when most of the organ systems become ready, and you will note this with sort of test or practice activities. So basically, the fetus will start to move, contracting its muscles. We will notice a phenomenon called fetal breathing. In fiber optics, you can see what will appear to be the fetus drawing a breath in. You can see all the appropriate movements of the chest cavity and the mouth during that time. But of course, it's not actually working in gas exchange. It's just kind of flexing the muscles that will commence breathing once the baby comes out. As I said, these parts, the respiratory system, is developing, and it is nourished and oxygenated by the activities of the mother's organ system, but is not providing any oxygen to the baby. It's breathing the amniotic fluid in and out. This is where the largest weight gain occurs, is that third trimester. And this is where the size of the fetus and the secondary tissues, the placenta and the uterus, begin to press against and displace many of the mother's abdominal organs. 4 months, you can see this definite human appearance with well-formed facial features. You can see the fingers with fingernails well developed. It's kind of interesting. Early on, these hands develop kind of like mittens with a single kind of sheath of skin. And then later in development, before this stage, enzymes develop, basically dissolve those connections to produce second fingers, separate fingers. So at 4 months, you can see the placement of the uterus, now at 4 months, kind of half size, with the fetus inside, still held low and well within the pelvic cavity, supported well by the bone structure around. But as that fetus begins to expand and eventually reaches full term, this fetus shown in an ideal position, head down. You can see the umbilical cord positioned so that the head can proceed out through the birth canal. Here is that cervix that we mentioned before, that muscular ring, which is retaining the mass of fetus and placenta and fluid inside this chamber, basically keeping everything together as it develops. You also notice from the size of the baby the reason for the soft wall in the abdomen. Up here above the liver and the diaphragm, you have a hard, bony cage protecting the thorax, the lungs, and the heart, big tube central, and the major ports of the lymph system. But down here, the development of this internal gestation, this mass of cells has to have a place to go. And where it goes, pushing back against the spine causes a flexing. And because we're bipeds, not at all convenient. This is very difficult for the female to basically maintain this postural alignment during late pregnancy, and lower back pain is very characteristic because of the posture. But the only place it can go is the expansion of this soft wall, both forward and outward and behind the screen as the skin expands against the pressure. The compression of the digestive organs shown here, down here below the compression of the urinary bladder and the lower end of the digestive tract, all part of late-term pregnancy. Now, the placental factors that have been sustaining the growth, I told you about the follicle as it is vacated. The follicle becomes the corpus luteum and sustains the pregnancy through its initial stages until the placenta reaches a size where it can take over that hormone production. So estrogens from the placenta are basically going to sensitize the uterine tissues, the myometrium, and as those increase, they make contractions more likely. As full term approaches, the development of the fetus activates stretch receptors basically throughout the skin and surrounding tissues, but also in the cervix. As that pressure increases, we see the sensitivity of smooth muscle fibers to oxytocin, which is a hormone from the hypothalamus. And that is a hormone that's associated with two reproductive factors. Labor contractions are accentuated, intensified, and the interval shortened by the presence of oxytocin. So it's important at the time of parturition. Oxytocin is also important in milk letdown. Prolactin is what initiates the milk production and its storage inside the breast, but it is oxytocin that causes that to be released along the ducts and deliver to the nipple. Fetal factors, basically the increase in weight and the distortion of the myometrium is what stimulates fetal development. The pituitary of the fetus releases oxytocin in response to the increasing estrogens. So late in development, we see this growing mass of fetus stretching the uterus and stimulating receptors all over the body. So we're going to get a response. Maternal oxytocin is going to stimulate contraction, labor pains. Prostaglandin production in the endometrium are going to stimulate further those smooth muscle contractions. And it's going to continue to press. The initial BRAC and HIT contractions are low tension and a short duration, but they are then repeated at higher tension, and the interval between them decreases until you have more and more pressure applied to the fetus inside the birth cavity. So eventually these labor contractions are supported and amplified. This is an example that we called positive feedback. If you remember the neuron picture, where the receiving neuron looped back and stimulated the cell prior to it in the loop, basically repeating and accentuating the loop toward the conclusion, in this case, the delivery of the baby. So before labor begins, this is ideal positioning, a so-called head-down position. The dilation stage depends on the relaxation of this cervical ring, so that it is typical at the beginning of labor. They will typically tell you 1 centimeter, and they want to see it at 10 centimeters before they begin labor and pushing the baby out. Finally, the main contraction point, the important thing is that for some reason, the development of the head in the human baby is disproportionate to the dimensions of the birth canal. It's typically the most difficult stage.